Good morning, everyone. My name is Hamid Ikhtiyari. I'm delighted to be here today. First of all, I put all my slides in a QR code. So if you scan QR code, you have the entire slides that I have. There are some unpublished data, so please do not publish them. But otherwise, everything is there. And before starting, I just wanted to thank Danny to make that happen. I mean, I was observing him kind of putting things together for something like 2 years, and I'm really happy that we finally had this chance to get together. So please join me in just thanking Danny for making this happen and bringing everybody together. So I don't have any conflict of interest. I should thank my colleagues and collaborators in the University of Minnesota and also at Laureate Institute for Brain Research that made those things happen. So I'm going to talk about transcranial electrical stimulation, talking about how that could be combined with fMRI or MRI in an online and offline way, talking about how we can do target optimization with TES, how we can measure dose responses. Also, I will talk a little bit about concurrent TES fMRI, the quality control, methodological checklist, everything about that. And at the end, I will talk a little bit about how to close the loop between TES and fMRI. So that is the final piece of the talk that I will present today and some next steps for the next levels. So as we discussed during kind of last day and today, we know that transcranial electrical stimulation, I mean, we have different modalities and how it actually looks inside the lab. We use multiple electrodes and we try to just kind of target different areas with electrical stimulation. One of the reasons I like the technology is it's really scalable, inexpensive technology. If it works, it's really going to be transformative in terms of providing some new interventions for people. And also it is moving towards what we call digitization. So we can just basically send everything in a package to our patients in home, and they can have everything there with a mobile app that is controlling everything. So even we can have our patients being in a trial without even seeing them for one. So that digitization is providing some interesting, exciting potentials. But why we are combining TES and MRI? What are the reasons behind that? One of the things that is pretty usual, and as Danny has mentioned and other speakers have mentioned, is about how TES is working. So just about the mechanistic understanding about how TES is working. So we do MRI before and after, and we have done some other studies in my lab as well in terms of how TES is changing brain network connectivities, or how we can use fMRI as an outcome measure in a larger scale trial, and some technical details about that. But beyond that, there are some potentials in terms of using fMRI as something like a predicting measure of response to TES. And also there are potentials for using fMRI as target optimization. So there are several potentials in using fMRI in combination with TES. When you think about integration of these two technologies, one way is basically what we call offline TES fMRI, doing fMRI before and after TES. And there are also potentials for what we call online TES fMRI. So we can basically we can do the fMRI while we are doing TES inside the scanner. If you ask me about the number of trials that we already have with these two types of modalities, as you can see here we have 160. It is updated by the end of February. So, I mean, we haven't updated that during last like 6 months, but it is almost that the numbers is not something like 10% more than this, but 160 trials with offline, and we already have 82 trials with basically concurrent or online TES fMRI. We have started to look into the technical details and parameter space of the trials that people have published so far. The challenging part is within those 242 trials, it's really hard to find two studies that they replicated something across different labs. So people are using various different methodologies, various different parameters. It's really hard to find two studies that has the same sort of protocol, which is a little bit kind of confusing, also frustrating. So we started to think about how we can bring people together and make a level of transparency in what we do and try to see if we can replicate the results that we have in different labs. So we started with that idea of making an international network. And we already have some like 54 labs across the world that are working TES, fMRI, and we try to put them together. I will discuss some of the outcomes of the collaboration as well. But in terms of using fMRI or MRI for target optimization, there are different potentials. Just one example, this is a trial that we are right now doing in terms of what we call frontoparietal synchronization. So we try to target frontoparietal network, and we try to synchronize that with alternating current stimulation. But we started to think about, okay, when we talk about frontoparietal network, as you can see in the brain, there are many parts in the frontal and many parts in the parietal cortex. And it's really hard to say, are we targeting the right part in the frontoparietal network? So we basically extended the modeling stuff, and we realized that we are interested to what we call drug cue reactivity, which is an important measure for drug addiction. And we realized that in the conventional montage that we have been using, we do not have any engagement of frontoparietal connectivity. There is no connection between F3, F4, F3, P3, and F4, P4 that we have been interested to. So we started to look more into details about, do we have any frontoparietal engagement with the cue reactivity task that we had? And we found, yes, there are frontoparietal engagements, but they are not exactly on the location that we have been looking for. So we realized that we basically need to change the montage from F3 to P3 to F3 to CP5, and from F4 to P4 to F4 to CP6. So we basically needed to optimize the montage that we had. And based on that montage, we have frontoparietal engagement. Then when we realized that previously we have been basically putting something like 2 milliamp in each node, and we had a kind of a reasonable distribution of electrical current, you can still see that there is a variation in terms of the amount of current that each individual would receive in frontal and parietal side, but they are pretty kind of equal to each other. But when we changed the montage, the new montage, we realized that then the distribution would not be equal. And if we want to have an equal distribution, we need to have something like 2 milliamp in one node and 1.2 in the other node. So this is how probably what we are using as MRI could inform the montage selection. We put that in something like a paper in brain stimulation. You can find some technical details about how to optimize montage selection for alternating current stimulation. And then there was a question regarding dose, how we can kind of adjust dose response measurement. In one of the pilot studies that we have been doing with fMRI and tDCS, we had significant reduction in craving in people with methamphetamine use disorder. But the interesting part was we had a significant inter-individual variation in response to tDCS, which we do not have in the sham part. And we have been thinking why we have such a wide variation in terms of the response. So we started to look into details, and kind of, as you can see, there are variations. So we tried to see if we can find a reasonable relationship between response and what we have in electrical field modeling. And we showed that, yes, there is a significant response between the peak of electrical current that each individual would receive and the behavior response that we had. We have done some initial analysis on that. We realized that basically the level of current that we have in the frontopolar area is related to the response that we have with fMRI. So we extended that in a larger trial with something like 60 patients with methamphetamine use disorder, and we started to realize that when we have basically electrical field modeling and we have fMRI, there are multiple levels of connecting dose to response, if we consider dose as electrical field and response as fMRI. So we can do that in the voxel level, we can do that in the kind of regional, we can do that in the active clusters, we can do that in the network level with multiple different variations that we can explore inside. It might be interesting that in the voxel level, we have not been able to find any sort of reasonable correlation between electrical field and fMRI response to electrical stimulation. And then we started to look into the kind of network level analysis, and we have some interesting results in the network level. So I'm not going to go to the details about that, but those who are interested, we are going to have a specific webinar, and this is a QR code if you are interested to join the webinar. It's a free of there is no cost for registration, and it's going to be happening in November 3rd and November 10th. So we are going to discuss about the concept of dose response in not only transcranial electrical stimulation, but all the other modalities of non-invasive brain stimulation. And we are right now discussing in terms of probably writing a framework about how to define these concepts and asking others to use the same terminologies or the same sort of levels of analysis to make a level of harmony inside the field. Going to the other level in terms of concurrent and online, there are some minor variations in terms of the terminology of concurrent or online. I'm using them as the same term here. But as I have mentioned, there are 82 trials that they did concurrent TES fMRI. And when we go to the technical details of how to do that, there are some various multiple details that people need to consider. So we started to think how we might be able to develop a steering committee of those people who are doing concurrent TES fMRI, and see how we might be able to develop a checklist, a methodological checklist for that. As you can see here, we already have some of the key figures already in the room. We have Axel, I can see Duke here. So many people who have been involved, they are already in this community. And we put the protocol in OSF, and we tried to see how we might be able to do it in a rigorous way. So we started to think that when we design this sort of checklist, it is not just me and my friends and friend of my friend who just get together in a kind of nice resort and discuss about kind of a checklist and we publish a checklist. So we have been thinking that how we might be able to do it in a systematic way. And we started basically to see what the people who basically publish things, we went into the kind of the publication. At that period of time, we had just 57 publications. So we invited those who publish in the field, we put them in an expert panel, we had multiple rounds of discussions, webinars, and we tried to kind of develop an initial version of the checklist, and then we asked people to rate the importance of different items inside the checklist, and we ended up to have what we call contest checklist. It has a short form and it also has a long form, and it comes in something like an Excel sheet, so you can use it as basically minimal items that every single concurrent TES fMRI study should report for the sake of transparency. And then we went into the previous publications and we realized that many of those items are really underreported in what we will publish before. And we think that all those items are important. If you want to replicate the study, we need to have those items to be reported in the study. So we are encouraging the community to basically to follow that checklist to be able to increase the transparency and also replicability in the field. And as you can see here, there are very few studies that they have more than 60% adherence to the checklist. So we hope that in 10 years we can come back and assess the adherence and hopefully have a better results in coming years. So we published that in Nature Protocols, and now that the package is available in Nature Protocol as a consensus statement. We are trying to basically bring others who are interested to collaborate. So that is kind of the collaborative effort of people who made that happen. Now we are working across different labs that are doing concurrent TES fMRI to see how we can set up a set of standards. Whatever lab you are working, before starting to do a concurrent TES fMRI, how you can collect some baseline data and make sure that the setting that you have would not make significant amount of noise in your data recording process. So we are defining something like a specific protocol, and we are asking other labs to just collect at least one set of data with this protocol and comparing the results that we have across different labs. This is an open call for labs that are interested to do TES fMRI, and we are hopeful to kind of have others be involved in this sort of quality control, noise control protocol. We haven't published the protocol yet, and it's open for those who are interested to contribute and be a part of the authorship team. We already have a YouTube channel that we put the videos and webinars and discussions that we have on that YouTube channel, so you can find us on YouTube and you can follow the network. Now, the last part of the talk about, is there any chance that we can close the loop between TES and fMRI? We do TES inside the scanner, and there are some targets that we are interested to. Is there any chance that we can have an online measurement of fMRI, of the target engagement, and loop that into the stimulation parameter and basically close the loop and optimize the stimulation parameter in each individual level? So that is what we are trying to do right now. To be able to do that, we started with setting up a setting in my lab at Laureate Institute for Brain Research, starting with watermelon, as you can imagine, and as you can see here, you can see inside the watermelon, controlling for the kind of the signal and noise and other things, and of course we eat watermelon after the party. And after watermelon, the next kind of thing that we put inside the scanner is, of course, the PI of the project should go inside the scanner and receive the stimulation. And that is the process that we basically started to move forward. We realized there are some challenges. We basically needed to make our own sort of electrodes to be able to kind of make that happen. Some technical details in terms of how to close the loop. But just a few days ago, we published a technical report on how to do that. And if you are interested to know the technical details, all those technical details and quality control, noise measurements, temperature measurements, all those things are inside that technical report, and you can go inside and find the details there in the technical report. And if you are interested in the concept, we put that in a concept paper. I'm hopeful that it will get published. So it's under review right now. And we have been successful in terms of collecting one set of pilot data, 20 participants, healthy control participants, in 2 wings, and we are doing data analytics with that. The thing that I can tell you right now, this is the kind of the side effects that we had in 2 groups. First of all, technically it was doable. So we have been able to put people inside the scanner, recording and trying to optimize the stimulation inside the scanner. And as you can see here, people have been pretty much blinded in terms of the effect, and the most significant sort of side effect was the sleepiness in both groups. I mean, the portable is pretty long, so that is something that I can at least tell you is technically feasible, but what is the right timing? What is the nice window of stimulation and recording and kind of optimization of stimulation? So there are many technical details that we can discuss further. Next steps. Right now, we basically submitted a new IRB application for doing that in substance users, and we are doing that while we are exposing them to drug cues, and we do that inside the scanner. So that's a new thing. Hopefully, if we get together in 1 year, we can show you some of the results. Final slides. We are realizing in the discussion that we probably need to go even beyond what we do in TS-fMRI and make a larger network. Something like we have been discussing with Danny in terms of what would be the right name for it, but we have been thinking about something like International Network of Neuromodulation Neuroimaging to promote virtual collaboration, diversity, global representativeness, transparency, and methodological rigor in the field. We made a WhatsApp group with few people that we are here, so we have started to kind of put people together. If you are interested, you can just scan the QR code in the WhatsApp group, and you can basically join the WhatsApp group that we made, and that could be a sort of a way for communication. We have started to think about putting together a few symposiums in HBM, and you know the time of the HBM changed this year to July, so it's going to happen in July. And they just opened the call for abstracts, so we are thinking to put some symposiums there. If you are interested, you are definitely welcome to join the symposiums that we are putting together for OHBM, and we have been discussing with Danny for potential for having something like study groups. So Danny is going to champion that, and hopefully that could be another potential in terms of bringing people together and make that collaboration possible. So take home notes. We discussed about potential for combining TS-fMRI, offline, online. We know that people are responding differently to TES, how we might be able to consider those inter-individual variations and take it into account to have better responses. We discussed that fMRI can potentially inform montage selection. We discussed about how to optimize dosing with head models, potentials for concurrent TS-fMRI, feasibility of doing concurrent TS-fMRI and closed-loop TS-fMRI, and potentials for multi-site collaboration. And this is again the QR code for my slides if you want to have the slides. Thank you very much for your attention.